Labbook.paper.
Exactly.
Version 3.
Actually, even worse than
Final.
that,
Alright,
so we're gonna go ahead and get started.
text files in various directories.
Thanks everyone for coming to Tools and Technology Seminar Series. There's a sign-in sheet going around. If you haven't signed in yet, please do so. So I'm pleased to present today's speaker, Wei Zhou, who is a recent graduate from the DCMB program, and she worked with Kristen Willer, I believe. And she's going to talk to us about a tool called SAIGE.
Hi everyone. Today I'm going to talk about this method I developed with my co-mentors, Dr. Sean Lee from Biostat department and Dr. Kristen Willer from the Human Genetics and Bioinformatics department. I'm going to start with a little bit of background on the genome-wide association studies. And as people start performing GWAS on large biobanks, and we have met some problems, and those problems drove the development of SAIGE. We know genetic variations can cause human diseases, and there are two main categories for human diseases. And the simple one we called Mendelian diseases. They are usually caused by genetic variations in one single gene. So those genetic variations usually aggregate within one or multiple families, so we can use family-based studies to try to identify those genetic variations. And on the other side, the other category of human genetic disease is called complex diseases. They are usually caused by multiple genes as well as environmental factors such as lifestyles. And examples include heart diseases, diabetes, and many other common diseases we see in the population. And so the genetic variations causing complex diseases usually have more higher allele frequency than the genetic variations causing Mendelian disease. However, their effect size for each single of them are relatively small. So the family-based studies which we used for Mendelian diseases no longer work for complex diseases. So the widely used approach to identify genetic variations for the complex diseases is a genetic association test. Here's an example to show how we perform genetic association tests to identify genetic variants that are associated with complex disease. So this example, we call it SNP1, and each of us have two copies for each SNP. And we call the two alleles, two different nucleotides for each SNP, two alleles. And the one with the lower frequency in the population, we call it minor allele. So for example, for SNP1, we have two copies, T and G. And we want to test whether this SNP is associated with a complex disease, for example, heart disease. What we're going to do using genetic association test is we're going to collect two groups of people, and one group containing people with the disease, we call them cases, and the other group containing people without the disease, we call them controls. So we're trying to compare the frequency of the one allele, for example, the G allele, which is the minor allele, in the two groups of people. And usually we use the logistic regression to perform the statistical test to test the difference for the G allele between the two groups. So here we see G is for the genotype of the copies of the G allele, and the X can include some other non-genetic covariates, for example, gender, age, etc. So when we expand the genetic association test to the genome-wide, and we have this genome-wide association study, we call it GWAS. And each GWAS can provide a plot like this, and we call it Manhattan plot. Each dot here represents a genetic variant, such as SNP, across the human genome. And the x-axis here is for their genome position from chromosome 1 to chromosome 22. And y-axis represents the negative log 10 p-value for the association, which we got from the logistic regression model. So the higher the dot and the more significant it is associated with the disease we're testing. And to correct for the multiple testing, and we usually use 5 times to 10 to negative 8 for the p-value as the significance threshold, and assuming there are 1 million independent genetic variants across the human genome. So as people starting perform GWAS in large cohorts, as large biobanks start collecting their participants, and they got information on a lot of phenotypes, a lot of diseases, as well as their genome information. So we can see usually a large cohorts have large number of participants and large number of genetic markers. For example, the UK biobank contains 500,000 participants, and for each of them, we have 71 million genetic markers genotyped or imputed. And another example I list here is the Hunt study, which is a Norway population-based biobank. And before the UK biobank, we think the Hunt study has the largest sample size among those biobanks that have been genotyped. So recently, last July, and UK biobank has released its data, the genotype and the phenotypes for to over 200 labs over the world. So we all have the data, and then we can all perform GWAS on this data. And the other feature of those large biobanks is it usually contains some related samples when they collect their participants. So here's an example to show there are about 55,000 participants out of these 70,000 participants in Hunt are related to each other up to 2nd degree. So we can see this large hairball showed us the strong sample relatedness. So we have to account for this sample relatedness, since if we treat each participant independently, and the inflated type 1 error rates can be caused. So here are more details for the UK biobank. And as we see, 1 in 3 of the 500,000 participants have at least one relative up to 3rd degree in the biobank. And here we filtered out some poorly imputed variants, and we have 28 million well genotyped or imputed variants for us to test. And since we have some questionnaires and also the hospital ICD codes for each participant, and we can construct over 1,000 different phenotypes, different diseases for us to test. So
what is ICD code?
Yeah.
ICD code was.
It's like international. It's like codes to represent different health record. Like when you see doctors, each time there is a code corresponding to like what symptoms and what situation happened for your doctor visit. Yeah. And if we use these ICD codes directly, and we will miss some other related information. So we have to have our phenotype expert help us construct some meaningful disease or phenotype for us to test, and by combining some related ICD codes. So usually people use linear mixed model to perform GWAS, and when we have related samples. So here, compared to the regular linear model, we have a random effect here, B, and B followed this multivariate normal distribution, and we have phi here, and phi is n by n matrix to represent the sample relatedness for each pair of sample. So you can see we have like 7 samples here, and we can construct a 7 times 7 GRM, which is this n by n matrix, and to show how closely each 2 samples are related to each other. Here is all existing linear mixed model that have developed for GWAS. And until 2015, the BOLD-MM was developed, and this one is the only existing linear mixed model method that can be applied to the large sample size, such as UK biobank. As we see, the N by N matrix here, it's a huge matrix to store. And that even it's symmetric, so you can just store half of it, and it still requires a lot of memory when we have large sample size like UK biobank. And also in the process, when we try to fit the model, we have to get the inverse of the N by N matrix. So to get the inverse of this large matrix, the computation time is big O to n to the third, so it's very slow. And so BOLD-MM is the only method that can be applied to the UK biobank. So all the other groups use BOLD-MM to perform GWAS in UK biobank. And we also tried BOLD-MM, but as you can see, for binary phenotypes, I show an example colorectal cancer here. If you use BOLD-MM and you see this Manhattan plot, and usually for well-performed GWAS, if we see some significant signals here, and we expect some specific spike, and not with a single dot here, and instead we're expecting multiple genetic variants exist for each single significant signal, because each genetic variant tend to correlate it with its nearby genetic variants. So as we see here, and there are a lot of scattered single dots here. So we see there are many false positives. And if you use BOLD-MM, perform GWAS on these binary phenotypes, and you decide, I have so many significant signals, and you decide to follow up these signals, you will waste a lot of time and energy.
How do you define control?
We exclude any samples with some related diseases, and exclude all of them, and then leave all the samples that do not have for example, colorectal cancer.
So remove any individuals with like disease related to the
Yeah, even those removed individuals, some of them can be defined as cases, but some of them just excluded from the whole analysis for each phenotype. So what we're missing, if we have used the linear mixed model to account for sample relatedness, and we're thinking about, should we really use linear mixed model for binary phenotypes? And even though the binary phenotypes does not really follow the normal distribution that the linear model assumed, and also linear model assumed the residuals have constant variances regardless of any covariate values, but this is really violated by the binary traits. So this can cause inflated type 1 error rates if we just use the linear model for binary phenotypes. As we can see, if you fit a regression line here, you probably have some problem. And back in 2016, the group, Shi-Hong Lin's group from Harvard has proposed this logistic mixed model. And so as you can see here, we use the logistic transformation for the probability for each samples for being a case. And we have the random effect here, just like a linear model. So we decided to use the logistic mixed model for the binary phenotypes. However, the algorithms they developed is very slow, and they take a lot of memory, as we expect if we take the large matrix. And if you try to apply this algorithm to one phenotype, you can course 184 CPU years, and for us to run one single GWAS. So we decided to use some optimization strategies to make this logistic mixed model work for the large sample size. Here what we have done, and to reduce the memory usage, we decide to only store some genotypes that we're using, try to compute the N by N matrix instead of store the matrix directly. So which means we don't store the matrix, and every time if we want to use we need the matrix, and we compute its element. So we only need to store the genotypes, and we can store the genotypes in binary vector, and so we can use 2 bits to store 1 genotype, because genotype can be either 0, 1, or 2. And so in this way, we save a lot of memory. We reduce this n plus 1 times 4, and if we store the whole matrix, we need this byte, and we only need n times m1 divided by 4. So this m1 is usually smaller than n. For example, we only have m1 equals 95,000 here. And so in this example of UK Biobank, we only need less than 10 gigabytes to store this matrix. And then, as we mentioned, to get the inverse of the n by n matrix, it's very, very time consuming. So we use this method called preconditional conjugate gradient to get the inverse of the matrix times some vector, and instead of use the Cholesky decomposition to get this inverse of the huge matrix. And in this way, we reduce the computation time. And then we also use some other, for example, randomized trace estimator to get a trace of the matrix of another large n by n matrix. Since we don't store this matrix directly, so we need this method to help us get the trace of it. And so in this way, we reduce our computation complexity from n to the third to m1 times n to the 1.5. And then we use these optimization strategies. We can apply logistic mix model for binary phenotypes. And this is the Manhattan plot when we do this. But we can see there are still many false positives here. Even we have used the logistic mix model instead of a linear mix model. So and we started thinking what is missing still. So there is another scenario here. When we look at the phenotypes in a large biobank such as UK Biobank. So you can see I plot here for the 1,663 binary phenotypes in the UK Biobank, and including those that have very small case numbers. And you can see the case control ratio is the x-axis, and the y-axis is the cumulative percentage of the phenotypes. And so if you look at this line, and you can see over 85% of them have case control ratio 1 to 100, which means the prevalence of this disease in the population is lower than like 99% or 1% in the. So which means there are very few cases versus a huge number of controls for each phenotype. And this can cause some problems when we perform the logistic regression or even linear regression. So because the test statistic will not follow normal distribution when the case control ratio is unbalanced. Here I plot the black solid line is the actual density plot for our test statistic when the case control ratio is 1 to 99, which means the prevalence of the disease is about 1%. And the dotted line is the actual normal approximation. So you can see our test statistic actually does not follow a normal distribution, and which may, if we assume it follows normal distribution in the logistic mix model, it can give us some problem. So we start looking at this method called saddle point approximation. It is initially proposed by Daniels back in 1954, and it can basically provide highly accurate approximation to probability density function for any distribution based on the moment generating function. And so instead, because we know normal distribution only use the first 2 moments, which is the mean and the variance, to define the distribution. But saddle point approximation can use the entire moment generating function. It can give more accurate approximation to the distribution of our test test statistic. So it actually has multiple applications such as bootstrapping and tail area approximation in other area. And so last year, the biostat student Renak, and in one of my co-mentor, Dr. Sean Lee's lab, has developed this method called SPA test, which try to correct for the inflated type 1 errors in logistic regression when there are unbalanced case control. So you can see the figure from his paper, like when we have balanced case control, and we even x-axis is the minor allele frequency for the tested genetic variants. So you can see for the very, very rare genetic variants, even the case control ratio is balanced, we see some deflation for score test, and the fast SPA is the test that he has developed, and the first test is another likelihood ratio test that have been developed to account for the unbalanced case control ratio previously. But it took the computation efficiency for this first test is very low. So, but you can see when the case control ratio become more unbalanced, and the type 1 error rates for the score test, which we usually used in the GWAS and combined with the logistic regression, got very inflated. And the SPA can correctly adjust for this unbalanced case control ratio. So we decided to also use this SPA and to add it to the logistic mixed model. Here, the method is only based on developed for logistic regression and not accounting for sample relatedness. So we add this saddle point approximation to account for unbalanced case control ratio and combine the optimization strategies for logistic mixed model. And we call this method SAIGE. And basically, our method contains two steps. And the first step, we use a subset of genotypes for subset of some hardcore SNPs and to construct to fit a null logistic mixed model. And so we need phenotype here and genotypes for those markers here. And after we fit a model, we got these parameters. And notice that this is the non-logistic mixed model, so we don't include any genetic effect here. And then after we fit the model for each genetic marker, we can use the parameters from the step 1 to perform the score test. And we apply the saddle point approximation to the score test statistic and get our association result. And so let's look at the runtime and the memory usage for SAIGE and compare to BOLD-MM. And the GEMMA is another linear mixed model method, and GMAT is the logistic mixed model method that without any optimization strategy. So the figure A here is for the CPU hours as the number of samples increased, and we can see the other GEMMA and the GMAT, the computation time is like dramatically increased, and this is by the log scale here. And SAIGE, which is the purple line, is in a similar scale as the BOLD-MM, and we see SAIGE still needs more time than the BOLD-MM. And we think the potential reason here is to fitting a logistic mixed model is harder than fitting a linear mixed model. And the memory usage, the same. SAIGE basically have very similar memory usage to the BOLD-MM, but the other two methods have very large, needs large amount of memory to fit the model. So, and we have performed GWAS on basically 39 million markers, include some other markers that you can use different minor allele count to decide which markers you want to test, and over 1,400 binary phenotypes in UK Biobank. And we use the Google Cloud to perform this, and we finished these jobs within 3 days. So this is the actual benchmarks, and we show here. And so for each phenotype, we only need 10 gigabytes for the memory, and 515 CPU hours. But you can see, if you can imagine, if you use GMAT, it will take forever to finish one GWAS. Here are some simulation studies I want to show, and so I simulate 1,000 families, and each family have 10 family members, like following this pedigree. And you can see when the case control ratio is 1 to 99, and if we use just GMAT, which is a logistic mixed model without SPA, and the inflation is more serious when the minor allele frequency is lower, and the SAIGE well corrected for these inflated type 1 error. And so as the minor frequency is about 0.5%, which are like less relatively rare variants in the population, and when the case control ratio goes more unbalanced, and type 1 error rates got more inflated, and the SAIGE corrected for these errors. So back to our problem, and we see this messy Manhattan plot with a lots of false positives for this colorectal cancer in the UK Biobank. And we apply SAIGE, and we see it's like very clean and well corrected for the type 1 error rates. And we have 3 significant signals, which are all previously reported by other GWAS. And then we randomly select other phenotypes. I'm going to show you 2 examples here, and one is the CAD. And so we see the case control ratio is 1 to 12, and we see this colorectal cancer case control ratio is 1 to 84. So the case control ratio is less unbalanced for CAD, and we see the blue dots are all previously reported signals, and green dots are all potentially novel signals because they pass the significant stretch line. And we still see some scatter plot here, and when we apply SAIGE, and we somehow clean the Manhattan plot. And another even more unbalanced case control ratio is the thyroid cancer, and we only have 358 cases in the UK Biobank. So the case control ratio is like about 1 to 1100 here. So if you use both MM, you basically got lots of false positives. And after we apply SAIGE, and we totally clean this problem, and we got one significant signal, and which is a well-known thyroid cancer signal. So basically, we have shown here our method worked for the binary phenotypes, and accounting for sample relatedness and unbalanced case control ratio. So this is the summary for this method. It does not require pre-computed kinship matrix because we only request for the genotypes for those genetic markers you want to use to construct a kinship matrix. And it's feasible for large sample size. It's developed for binary trace and accounting for unbalanced case control ratio. And we have also implemented the function, the linear regression mixed model in SAIGE. So you can also use it for testing quantitative traits. And we compare this for GMAT and BOLD-MM. So we basically add like one dot here, and for people to use to perform GWAS. So the code is available as an open source R package in the GitHub for people to use, and we have released all of the GWAS results to the public. So it is freely freely for the public to download. And if you want to look at any GWAS results for any of these binary phenotypes in UK Biobank, you can go to download it. And we have also include these results to the Michigan FieldWeb, which is like web server for people to browse those GWAS results, and then you can click on any dots and go to see more higher resolution results, and by look at the regional association results. So here, this work is a collaborative work, and it is supervised by my co-mentors, as I mentioned, Dr. Sean Lee from the biostat department, and Dr. Kristen Wheeler from the bioinformatics department, and also Gonzalo Abcasis and Han Yun, and have like we've been worked together to develop this method. And I want to thank all of the members in the Wheeler group and Shang-Li's group and Abcasis group and my this is the method. Thank you.
So I actually have a question.
Yeah.
If you go back to the slide with like the thyroid or the case control graph, so something like this.
Yeah.
So you point out that in SAIGE it comes up with the known loci.
Yeah.
But do you know are there other known loci that SAIGE is not picking up?
Yeah, of course, there are more than one known thyroid cancer loci, and I think it's probably because I'm not sure if the case number is very low, so we probably do not have enough power to pick up all the other known loci.
So another question is, when you compare to other methods, their p-value is over-optimistic, but does not mean that the ranking of the p-value doesn't make sense, right? Maybe they just make the p-value too small. So, is there any way like precision recall curve or something that shows that?
Yeah. So here I have some backup slides for to show the power. So we have done some simulation study to evaluate the power of the SAIGE and compare to both MM. So basically, we have tried different case control ratio and different odds ratio here, along with different myelin frequency for the markers. So the green line here is for SAIGE, and the red line here is for kind of a SAIGE no SPA, which is equivalent to GMAT, and then the other lines are both MM. So you can see when the case control ratio become more unbalanced, SAIGE have more power, but it's basically because they have the other two methods have very, very inflated type 1 error rates, and so we
here is all the tests done at a fixed p-value cutoff.
No, we use different empirical p-value cutoff, which is corresponding to the type 1 error rates 5 times 10 to the negative 8. So so the main reason here is for the other two methods, because of the inflated type 1 error rates, their significant cutoff is more significant, like more stringent than SAIGE. But yeah. Yep.
Could you go back again for the thyroid? Like with this, like 300 cases and then 400,000 controls, can you give us an idea about like what kind of a sample size a data set with 1-to-1 ratio of case control equivalent to that, like for the power?
Right. So I guess you can usually we expect we estimate the effect sample size. Yeah. So the effect sample size equals 4 divided by 1 minus cases plus 1 minus controls. And so I can imagine the effect sample size, like when the case control ratio goes over like 1 to 100, increasing more controls probably contribute little to the power of your test. But I think the advantage here when you have more controls is you got to detect more rare variants. And I have done some evaluation when you can just forcibly make the case control ratio 1 to 10. And in this case, you have about less than 4,000 total samples. And you can see when you basically cannot detect those very, very rare variants in the samples. So I can Here I have a QQ plots for, so this is the thyroid cancer QQ plots for the 3 methods we're trying to compare. And both MM and SAIGE without SPA is equivalent to the GMAT, and this is SAIGE. And the dots are stratified by myelin frequency. So you can see when out of the 28 million tested markers, we use myelin count larger than 20 here to filter out those very, very rare variants. So about 90 million of them have myelin frequency lower than 0.5%. And which is represented by the gray and the black dots. So if you only kind of decrease your case number, and you probably will lose lots of these rare variants since they are not existing in a small sample size. Yeah.
Can you go back to your appendix slide on of complexity?
Yeah.
Okay. Yeah, that's pretty good in terms of. Do you think you can improve it more down the line? Like get it to just be straight linear for all steps?
Yeah, actually here the 0.5 here is because the conjugate gradient algorithm to get the inverse of the n by n matrix is a iterative matrix.
Yeah, that's a very inefficient step.
Yeah,
if you can avoid doing that in the future, you can improve your program a lot.
Yeah.
Is the 0.5 an empirical value, or did someone actually?
It's an empirical value. Yeah.
Yeah, I don't think I've ever seen 1.5.
Yeah, since both MM method has used this use PCG for the matrix inverse, and they have plot their plots and to show it's like equivalent to n to the 0.5. Yeah.
So if anyone's going to do fix that step, get it down to straight linear, would it be you or would it be someone else working on it down the line?
I think probably someone else. Currently we're extending this method to a gene-based method. Yeah, to make scale accounting for sample relatedness for the large-scale data.
Your thyroid cancer data said there are variants, there are known variants that you didn't detect. Did you actually look to see if in your sample that they exist?
You mean those rare variants?
Yeah. It'd be an interesting thing just to see, like, is it in my population? Am I missing it because of or it's just I don't see it at all?
You mean sorry.
I'm not trying to detect it statistically. They're known, so you should be able to look in there, yes?
Yeah, yeah. They should exist in a sample, so that's why we can compare their allele frequencies in cases and the controls.
Do you know that the other known variants exist in your data?
In UK Biobank, you mean?
Whatever you're using.
Yeah, in UK. I haven't go to look at the individual level data, but what I mean is here you mean other known signals or I haven't checked them, but yeah, I think it is as long as the frequency of those signals are
we don't know. You could actually look in the data.
Yeah. Yeah.
It'd be an interesting thing to see. Are they really rare in my sample, or am I missing it for some reason?
Yeah. What I did is I plot the QQ plot stratified by all variants and removed any variants around the known signal, not only for this one known signal, around any known signal previously reported, and we see the stratification. We see the signals that around other known signals also have you can see their p-values tend to higher than
random. And also is getting to somewhat the ranking question, because
Mhm.
you may be able to rank some other things if you're not filtering it out.
But what I'm thinking is, given all of these messy signals, even those known signals are ranked on the top of these messy signals, and it's hard for us to determine whether they're true or not.
Great, thank you.